Stem Medicare, noble care for human nature. Retrogenesis therapy with immune-tolerized extracellular vesicles. In a healthy state, our cells properly regulate gene expression, synthesize proteins, and carry out metabolic processes. However, internal and external stressors, including inflammation and infection, can impair vital cellular functions. As DNA damage accumulates and telomeres shorten, cells undergo aging. These processes constitute the hallmarks of aging. A core function of cells is to synthesize the proteins they need for survival. The first step is to create messenger RNA, the blueprint for a protein, in the nucleus. In the cytoplasm, mRNA from the nucleus is assembled into a polypeptide chain by ribosomes and transfer RNA. This process is called translation. A particularly serious problem is the disruption of protein homeostasis, which is primarily regulated by the proteasome and autophagy. Proteasome is an intracellular organelle that degrades misfolded proteins into amino acids. However, when misfolded proteins increase and form toxic aggregates due to various factors, proteasomal degradation becomes more difficult. These toxic protein aggregates can be removed by intracellular recycling system, called autophagy. Autophagy protects the cell by clearing out accumulated trash, such as misfolded proteins and damaged mitochondria. The process begins when these waste materials are captured within an autophagosome, much like being placed into a trash bag. Next, this joins with a lysosome, which is packed with digestive enzymes. Inside, the waste is broken down into basic building blocks, like amino acids and fatty acids, which are then recycled as new energy sources for the cell. Proteasomes and autophagy are essential functions for maintaining intracellular protein homeostasis. However, if protein balance is disrupted due to aging or stress, toxic proteins accumulate, leading to neurodegenerative brain disorders such as Alzheimer's and Parkinson's disease, as well as various systemic diseases. If these toxic protein aggregates leak out of cells and accumulate in blood vessels, they can cause vascular diseases such as atherosclerosis. Then, is it possible to restore aged cells back to a healthy state? It is possible if the disrupted homeostatic signals in aged cells are normalized. We have newly developed this treatment paradigm under the concept of retrogenesis therapy. Retrogenesis therapy refers to a new therapeutic paradigm that restores a healthy state prior to onset or aging by maintaining cellular homeostasis, as follows. The first is the maintenance of intracellular protein homeostasis through the normalization of proteasome and autophagy functions. The second is the maintenance of extracellular protein homeostasis through the normalization of phagocytic function by macrophages. The third is the maintenance of intracellular signal homeostasis through the normalization of intracellular signaling, such as between immune cells and stem cells. The fourth is the maintenance of stem cells' stemness. Homeostasis through the activation of self-renewal in aged stem cells. The key of retrogenesis therapy lies in extracellular vesicles from fetal stem cells, which possess all the signals necessary to normalize biological signals disrupted by various stresses. We have designated these as MBTCEVs, signifying that they contain the code to normalize malfunctioning biosignals. MBTCEVs are nanoparticles derived from early pregnancy of fetal stem cells, which are the youngest stem cells that can be obtained without ethical issues. MBTCEV contains over 9,000 types of proteins, over 50,000 types of mRNAs, and over 2,500 types of microRNAs, which is much more than typical adult stem cell-derived EVs. These components are delivered directly to damaged and aging cells via immune-tolerant EVs that do not trigger an immune response, performing various biological functions such as blood vessel and nerve regeneration, regulation of inflammatory responses, and cell migration and differentiation. The true power of MBTCEV lies in its ability to reactivate the cellular powerhouse, the mitochondria. It has been confirmed that MBTCEVs can increase mitochondrial DNA expression in target cells by up to 25 times. There is interesting scientific evidence to support our findings. According to a recent study published in Nature Aging, young EV derived from young mice demonstrated that it can enhance mitochondrial DNA expression throughout the body of aged mice, including the hippocampus, heart, lungs, and muscles. These anti-aging effects also revitalize our organs. For instance, young EVs restored aging heart function to youthful levels. The benefits also extend to bone health. Young EVs reversed aging-related bone loss, restoring bone density. Similarly, young EV successfully prevented brain shrinkage and volume loss. Furthermore, these organ-level anti-aging effects induced by young EV demonstrated remarkable results, ultimately extending lifespan by more than 12%. This is the beginning. Brain, heart, liver, cellular. It can be applied to any age-related disease where protein homeostasis has been disrupted. While time cannot be reversed, health can be restored. A science that repairs disrupted signals, restores cellular balance, and redefines the timeline of life. This is the retrogenesis therapy. STEM Medicare, noble care for human nature.